Krebs cycle or TCS cycle, that is tricarboxylic acid cycle. In continuation of glycolysis, the pyruvate which is generated from the glycolysis reaction is entered into mitochondria for the TCA cycle. The TCA cycle is otherwise called citric acid cycle. This is named after the formation of initial product citric acid. So this is the respiratory cycle which take place in mitochondria, and this cycle is named after the scientist who discovered the cycle. He is a biochemist, Hans A. Krebs. That's why it is otherwise called Krebs cycle. The pyruvic acid is converted to acetyl-CoA in the presence of enzyme pyruvate dehydrogenase. This is the initial step and it enters into the Krebs cycle. The first step is the condensation or formation of citrate. Acetyl-CoA is converted to citrate in the presence of enzyme citrate synthase. The second step is isomerization. There is a formation of isocitrate from the citrate molecule in the presence of enzyme aconitase. Third step is second oxidative decarboxylation. Here there is a formation of alpha ketoglutarate. Isocitrate which is converted to alpha ketoglutarate in the presence of enzyme isocitrate dehydrogenase. The fourth step is third oxidative decarboxylation. Here alpha ketoglutarate is converted to succinyl-CoA in the presence of enzyme alpha glutarate dehydrogenase. Fifth step is the synthesis of ATP GTP. The succinyl-CoA is acted upon the enzyme succinate or succinyl-CoA synthetase thiokinase, that is the enzyme succinate or succinyl-CoA synthetase thiokinase, form 4-carbon compound succinate. That during that time there will be a release of GTP, that is guanosine triphosphate. Sixth step is dehydrogenation, oxidation of succinate. In this step, 4-carbon compound succinate is dehydrogenated into 4-carbon compound fumarate in the presence of enzyme succinate dehydrogenase. Here FAD is converted to FADH2. Seventh step is hydration of fumarate. Here the fumarate 4-carbon compound is converted to malate in the presence of enzyme fumarase. Eighth step is oxidation of malate or dehydrogenation of malate. So the malate is oxidized or dehydrogenated. Here in the presence of NAD, it is converted to NADH. During this process, malate is converted to oxaloacetate. So this is the final step of the Krebs cycle, that again which is converted to citrate. The reaction will started from pyruvate and it directly synthesize oxaloacetate in one way, and pyruvate to citrate in intermediately the synthesis of acetyl-CoA, and after that it will convert it to citrate. So this is a Krebs cycle or citric acid cycle. Here 2 molecules of GTP is generated from the pyruvate molecule. As like as ATP, GTP is also an energy molecule. In addition to that, 6 molecules of NADH and 2 molecules of FADH2 is synthesized. So this is about the Krebs cycle.